Hi, my name is Julie Alexander. I'm a postdoc researcher with Oregon State University, and we are doing research on the Klamath River in California and Oregon.
And I'm Michelle Jordan, and I'm a master's student at Oregon State University as well, working on a similar project as Julie is.
The problem that we're actually focusing on is we're trying to better understand the ecology of this tiny little worm that actually really looks like a little Rastafarian. It's about 3 millimeters tall, so it's really tiny, and it's got these crazy tentacles that are feeding palps. That's how they eat and breathe, and they look like dreadlocks. But the thing about these worms is no one really understands them. We don't know a lot about them. We really don't know where it lives, why it lives where it lives, and so we're actually out there diving in the river trying to determine what sort of environmental factors are conducive to allowing this worm to proliferate. Things like environmental temperature and pH are really important to invertebrates because they can't actually regulate their own body temperature. They can't control the chemicals in their environment. So if you can actually figure out which factors are important to their life cycles, you can sometimes pinpoint where they might be in a particular system. A long-term goal for the findings of our research will be to help us actually pinpoint potential areas of this worm's life cycle that we could then interrupt the parasite's life cycle. So this worm actually becomes infected with the parasite. The parasite changes body form inside the worm and then is re-released back into the environment where it can then attack and infect the salmon. It then changes form again and reinfects the worm. And because the worms are more stationary, they can't move around like a fish can, we are really interested in trying to figure out particular aspects of the environment that might limit the worm's population, because that could then limit the parasite's habitat. You limit the worm, you limit the parasite.
My master's thesis is related to what Julie's studying. Julie's more focused on the ecology specifically, and I'm looking more at the physical habitat. So what we want to do is actually be able to model how much of the habitat of the polychaete is actually in the river. And the way that we can do this is there are these really brilliant people that have created these awesome computer models where you can basically build your river on a computer and you can model what's happening. So you can say, I have my stretch of river and we have 3,000 cubic feet per second of water flowing through it. What are the depth and velocities at all these different parts in the river? So what we want to do is take this model and say what's going on in the river, and then say which of those depths and velocities are suitable habitat for the polychaetes. And then from that, we can say, we can make predictions about what types of flows in the river create the least habitat for the polychaete. And again, like Julie was saying, if you can decrease the amount of habitat for the polychaete, then you can decrease the numbers of the polychaete and hopefully decrease the amount of parasite that the polychaetes are releasing into the water and help the fish out. Right now, what we're doing is amassing a lot of biological data on where we're finding the polychaete. So we're taking samples of the polychaete and we're measuring depth and velocity, and then this information will help us to model polychaete habitat. The Klamath River is a dammed river, so what this means is that the water that's flowing downstream of the dam is regulated, and what tends to happen in a dammed river is that the flow is less variable. So in a natural system, what will happen is in the spring a lot of times, or in Oregon in the winter due to rains, you get a peak in the flow, and so you have really high velocities and a lot of water flowing through the river, and then as summer comes along, that dies off as things start to dry out. And so this is known as a dynamic system. And when you put a dam in, what tends to happen is that you have a less dynamic system, and so you have a more steady flow rate during the year. Now, one of the hypotheses is that in the Klamath River, since it's a dam system, that the flow is less variable now, and now that it's regulated, and that what is happening is that the polychaetes have more habitat now, because previously when there was a more dynamic system, the high flows in the springtime would have flushed out some of these areas where the polychaetes were living, and now they can stay established in these areas in bigger populations.
So a specific focus of our research is actually trying to better understand the polychaete life cycle. Specifically, we don't know what happens to the polychaetes over the winter. We know that as soon as temperatures and daily light regimes reach a specific level, we see a population explosion. Numbers grow, densities are much, much higher, and then towards the end of the summer they appear to drop off again. So we assume that recruitment, progeny production, is really high right around July, August. And basically what we think is going on is that as soon as light, temperature, and potentially even food availability reach certain levels, the polychaete appears to be able to take advantage of that and absolutely explode. They go from being almost undetectable to being present in these unbelievable shag carpets of polychaete colonies. They're everywhere. I mean, you look down, you can't miss them. It's pretty impressive. So one of our questions is, there's this huge dichotomy of how is it that we are detecting these huge peaks of the parasite in the spring when the fish are moving around and the population of the polychaete is still so low that we actually can't even find them. So what we're seeing are two different peaks, essentially. There's the parasite peak that makes it look like the parasite has achieved its highest densities much, much earlier in the spring than we see the polychaete achieving its similar densities. So we're not really sure how this is matching. This is a great mystery associated with this particular disease, and understanding the parasite's life cycle and actually marrying it to not only the polychaete's life cycle but also the fish life cycle. How this disease works is, it basically comes down to three concepts: the parasite's life cycle, the polychaete's life cycle, and then the salmonid life cycle. So we know and understand a lot more about the salmonid life cycle. We know when they are moving up the river, and we know when the juveniles are moving down the river and are potentially being exposed to the parasite. And what we know about the parasite is that it appears to achieve highest numbers in the early spring. That's when, in our water samples, which you probably heard about earlier, we actually detect the parasite in really high levels, and this is in early spring. We detect the polychaete in the highest levels or highest numbers actually not until much, much later in the summer. And so we're having a lot of trouble actually coupling the parasite's life cycle to the polychaete's life cycle. And we're not really sure how these two different pieces of the puzzle are integrated yet. So this is something that we are actually focusing on. We are doing year-round sampling, or we're going to attempt to do year-round sampling this year, in order to actually see if we can track the population of the polychaete through seasons and... Finish my sentence for me, that's what you do.
Figure out where they're hiding out.
Yeah.
Yeah. Where's their safe house? Their polychaete
Right.
safe house.
Once we have taken these polychaete samples, we bring them back to the lab, we look at them under a microscope to make sure that we've actually got our little Bob Marleys, and we then take Bob Marley and we shove it in a tube, extract the DNA.
Grind them up.
Yep, grind them up, extract the DNA, and then we test their DNA for parasite DNA. So the parasite is even smaller than the worm, so you absolutely, unless you were really motivated, it'd be very hard to actually see the parasite in the worm. So we use these really elegant molecular tools to just detect the parasite DNA for us.
Polychaetes are great. There's a lot of job security in polychaetes because
no
they're
one
not
wants
cool.
to do it.
They're tough.
hard.
It's hard.
Yeah.
It's cold in the water in December.
So during our monthly sampling, we actually go out and we target specific physical features in the river to take our biological samples, our polychaete samples from. We have eddies, we have pools, and we have runs, which are all common physical features in rivers. We dive down, we collect the polychaetes, we come back up, we take down some physical habitat gear, we measure various aspects of the habitat. We measure things like substrate, different water chemistry conditions. These are all important factors for the polychaete. We take these biological samples, we head back to the laboratory, and we actually take Bob Marley, we check him out under the microscope to make sure it's really him, and we then use all of these really elegant molecular tools to determine whether or not Bob Marley is infected with the parasite. So this actually involves taking the little Bob Marleys, putting them into individual tubes, and these guys are really tiny. You have to sit under a dissecting microscope, and then you head over into another laboratory where you actually run polymerase chain reactions to detect parasite DNA. And this is a pretty elegant tool because the parasite is so small, it's inside this already tiny worm, so it's really hard to detect visually. So what we end up doing is we just extract the DNA, and then we test the worm's DNA for parasites. And so this makes our lives infinitely easier. For every hour of field work that we do, I would say we have another 40 in the lab associated with processing it all. So it takes a special sort of focus.
And the strains are interesting because they seem to have different virulences associated with the different species of fish. So one strain is more virulent for, say, coho, and another is more virulent for chinook. I think field biology is some of the most fun science ever. It's also great to get back into the lab and do things in a controlled environment where you can manipulate everything, but as a field biologist, you never know what's going to happen and what's going to go wrong, and you have to really think on your feet, and that can be fun and exciting. Drew and I have talked about how we expect to get honorary engineering degrees out of this by the end of our projects, because every day something new breaks and you have to figure out some new way of fixing things. You know, the field biologist's best friends are duct tape and zip ties.
We're often 4 or 5 hours away from hardware store or any sort of help. So we have to be able to use tools that either won't break, or that when we break we can zip tie them together and continue as needed. So Jerry Bartholomew, whom we both work for, seems to not only be an excellent biologist, but she's a really good judge of character. And so this is useful, obviously, in any sort of supervisory role, but we tend to benefit from it pretty well because all of the people that we end up working with in the field work together pretty well. There have been no major dramas. Certainly if a camera crew came with us, I don't think they'd be too impressed with us because there's not a lot of real-world drama going on in it. Usually people have different strengths and different weaknesses, and she seems to recognize that and make sure that she integrates her teams well. A common feature of field biologists is that many people perceive them as dorky, but we think it's just more that we've developed a more wacky sense of humor. Like Michelle has developed this polychaete dance. You know, and people think we're pretty weird, but it works. It keeps us from murdering each other in the field, and it allows us to collect the data that we're out there trying to collect.
You also get to do things sometimes that you would have never expected that you would. For example, for this project, we were actually scuba diving, and I got to get scuba dive certified for this project. And so we're diving in the river, which is really cool, and you get to see things that you wouldn't have gotten to see otherwise.
It has actually opened up some doors for us because, as a result, both of us are on this list of university certified research divers, and you can imagine how this will eventually, people get into a tight spot, they need an extra researcher. Oh, look, here are Michelle and Julie on the list. They're a little weird, but let's bring them along.
Looks good on your resume,
yeah,
too.
Science doesn't have to be all about calculating differentials or anything like that. There can be some fun activities associated with it.
It's really neat with science, too, to feel like you're contributing something. We're getting to take a little piece of this puzzle and attack it from our point of view and try to solve it, and that piece of the puzzle ultimately will be able to go to help solve a problem, a real problem, and that is a really cool feeling to get to do, and to also get to find an answer to something. There's a question that there is no answer to. You spend a lot of your young education all the way through undergrad learning things that people already know and memorizing things that people have already discovered, and now you get this opportunity to figure out something that no one else knows, and that is a really cool thing, I think.